Next, we're going to look at VLDL, LDL, and IDL lipoproteins. VLDL is produced by the liver. Its purpose is to transport fat from the liver to other tissues of the body. We will consider the transport between the liver and adipose tissue only. So as you can see, when fat enters the liver is going to take that fat, utilize it, and release some of that fat in lipoproteins called VLDL, very low density lipoproteins. VLDL carries triglycerides and also cholesterol esters. The structure of VLDL is similar to chylomicrons, except that it uses a different integral protein called ApoB100. Chylomicrons had ApoB48, VLDL ApoB100. Both ApoB100 and ApoB48 are produced by the same gene. VLDL travels into the blood, and within the blood, just like chylomicrons, it obtains two new apoproteins shown here. And again, they are ApoE and ApoC, which it obtains from HDL, high density lipoproteins. The chylomicron then travels to the adipocytes, and just like in chylomicrons, it's going to bind two enzymes on the adipocytes called lipoprotein lipase, or LPL, using its ApoC. When it binds to the lipoprotein lipase, this enzyme is going to cleave the triglycerides in this VLDL. It's going to cleave the triglycerides into individual fatty acids and glycerol, and the fatty acids and glycerol are going to be absorbed into the adipocyte as shown here, free fatty acids and glycerol. So as long as VLDL is interacting with lipoprotein lipase, the level of its triglycerides are decreasing as they're being broken down and absorbed into the adipocyte. When the level of triglycerides is reduced to about 50%, the VLDL unbinds and travels back to the liver. And within the liver, it's going to bind to the LDL receptor using its ApoE, and will be uptaken by the liver, recycled, refilled with triglycerides, and released back into circulation. However, if the VLDL does not unbind, or if it unbinds and rebinds to another lipoprotein lipase, in other words, it does not unbind and go to the liver, but rather is continued to be used by adipocytes, the level of its triglycerides is going to continue to go down. So shown here. So see here, the VLDL remains bound to the LPL, and therefore its triglycerides continue to be reduced. When its triglyceride level reduces to about 30%, the VLDL is now called IDL, intermediate density lipoprotein. So IDL has less triglycerides than VLDL, but it has more cholesterol esters than VLDL because there is a decreased percentage of its content as triglycerides, and therefore increased is cholesterol esters. IDL can then go to the liver and via ApoE bind to LDL receptor, be uptaken by the liver, refilled, and released back into circulation in the form of VLDL. If the IDL does not do that, but rather continues to interact with lipoprotein lipases, again, the level of triglycerides within the IDL is going to continue to decrease. So now, when the level of triglycerides within the IDL is reduced, let's say to about 10%, the IDL loses both ApoC and ApoE and is now called LDL, low density lipoprotein. So the structure of LDL is similar to the structure of VLDL when it was first released from the liver. It contains a single ApoB100 and no peripheral apoproteins. However, the difference between this LDL and that VLDL here is that there is very little amount of triglycerides within LDL, and therefore more of the LDL is occupied by cholesterol esters. VLDL had a lot of triglycerides, and therefore less of its core was formed by cholesterol esters. But as we saw, as VLDL continued to interact with lipoprotein lipase, its triglyceride levels reduced to 50%, then to 30% IDL, and then IDL further interaction with lipoprotein lipase forms LDL, only 10% triglycerides. So the level of triglycerides is reduced as you go from VLDL to IDL to LDL, whereas the cholesterol content is increased. LDL is then taken up by the cells in order to use its cholesterol. In other words, LDL, you can think of it as the major source of cholesterol in the body. So when cells need cholesterol, they're going to take up LDL. So we said that LDL lacks ApoE and ApoC. This means that when the LDL, or low density lipoprotein, travels back to the liver, we said previously that lipoproteins are taken up by the liver when they bind to this LDL receptor using ApoE. So if LDLs do not have ApoE, they cannot be taken up by the liver. However, ApoB100 can bind the LDL receptor. However, the binding, the affinity of ApoB100 for the LDL receptor is very low. So this means that the half-life of LDL in the blood is much longer than IDL and VLDL. It is usually stated that LDL is bad cholesterol. However, that is a misnomer. That is wrong. LDL is not bad cholesterol because LDL is the source of cholesterol for the body, and cholesterol is very important. Cells use cholesterol to produce cell membranes, to make cell membranes, as well as to make steroid hormones such as cortisol, testosterone, estrogen, aldosterone, and so forth. So cholesterol is very important for our body, and LDL being the major source of cholesterol is very important. So it should not be called bad cholesterol. It is called bad cholesterol because when LDL is present in high amount, when its levels in the blood is very high, then the LDL becomes a problem. And the reason is because LDL can get oxidized by free radicals in the blood, and when it gets oxidized, it's going to bind to the endothelium of your blood vessels. So there's the endothelium, and it's going to deposit the cholesterol in just beneath the endothelium, and then this leads to atherosclerosis, hardening of the blood vessels, as well as formation of pus, and platelet aggregation causes narrowing of the lumen of the blood vessel, leading to problems such as heart diseases and stroke.